Well, welcome everyone to the University of Maine Systems Research and Innovation Speaker Series, where we are featuring research and development projects, programs from around the system that are supported by internal funding mechanisms such as the Maine Economic Improvement Fund, the UMS Research Reinvestment Fund, and other really impactful experiences that have been funded also through external funding opportunities. This monthly series is a great way to highlight the incredible research and programming that's being conducted across the state of Maine. Today's speaker is John Muthyala, professor of English at the University of Southern Maine. He leads the Digital Humanities Initiative, served as the chair of the English department, and was PI of 2 Maine Economic Improvement Fund grants, Digital Maine and Culture, Commerce, and the Environment, Iceland and the North Atlantic. Oh, those sound exciting too. I'm really interested to see where which directions we're going to go today. His teaching and research areas are digital humanities, globalization, and international American studies. The digital innovation in liberal arts demonstrates productive synergies that can result from collaborating across disciplines to engage in digital innovation. He's currently working on a scholarly monograph on the impacts of drones and surveillance on American foreign policy, society, and culture. It draws on the discourses of digital humanities, post-digital cultures, and the new aesthetic. Fascinating. Well, today, maybe if there's time, we can hear about some of that other really great work that you're a part of. But the presentation itself is in reference to an NEH, National Endowment for the Humanities Summer Institute experience about why bioengineering needs the humanities. I'm just really excited to hear more about this. John has asked that at the beginning, he's going to give us a general overview of the experience and lessons learned and such. And then we can have a conversation at the end. So if you have questions, write them down on a piece of paper or put them in the chat, and we can address them once John has given his overview. So with that, I will hand the mic over to you, John. Thank you so much for being with us today.
Thank you, Danielle. Listening to you, I'm thinking, I think I have to record that and play it over again. That's such a nice introduction. It's like, it's like, wow, do I recognize myself there? But thank you for that generous introduction. Thank you to Jason Charland for the invitation, and to Tammy and Ed and Danielle and anybody else who was involved in putting this together. As Danielle mentioned, I attended the National Endowment for the Humanities Institute this summer at Arizona State University, where we had scholars from across the country meet for an entire month for intense study on the topic bioengineering, ethics, and the humanities. The co-directors were Jason Robert from the School of Life Sciences at ASU and Dr. Erica O'Neill from the Lincoln Center for Applied Ethics. So my talk is not going to be a report of what we did so much as my reflections and the way in which we can probably extrapolate from that work to make connections to what I'm doing, and hopefully to make connections with so many other scholars are also doing in the University of Maine system. So we probably won't be taking a long time. I'll speak for about 20-25 minutes or so. I want to make sure that I get some key issues out, and I'll be reading this. I don't have a PowerPoint presentation. And then we can have a Q&A. And I'm doing this to generate a conversation, not to present on a research agenda or a project or even a pedagogical idea. Although all of those can come into play. It is primarily to start a conversation, to put into practice and affirm what Einstein said quote him, all religions, arts, and sciences are branches of the same tree. All religions, arts, and sciences are branches of the same tree. And we as professionals are so used to differentiation and hyper-specialization. We are socialized as professionals in our fields, in our professional organizations. We get tenure, we get hired, we don't get tenure, we are sometimes fired for not doing what the discipline, so to speak, expects us to do. So it sounds like a platitude when we say the religions, arts, and sciences are branches of the same tree. But to understand how those branches come about historically, and to make those connections, and to understand how this tree of knowledge, in a sense, functions, it is a very difficult intellectual endeavor. So I speak here with a lot of hesitation. I probably am going to mess up a couple of words that I'm not going to be pronouncing well, so I apologize in advance. And if there are questions that you ask, and if I say I have no clue how to respond to it, but we will have to refer to our colleagues in the humane system, that's probably what's going to happen. The talk is titled, Why Bioengineering Needs the Humanities. Why Bioengineering Needs the Humanities. This July, Rob Toews published an article in Forbes magazine titled, The Next Frontier for Large Language Models is Biology. The next frontier for large language models is biology. The title intrigued me, and I read the essay, and also because of our fascination and our perplexity with OpenAI's ChatGPT, which uses large language models. What Rob Toews says is intriguing, and I'll read a couple of sections from his essay. He says, one striking theme has emerged from the long march of progress across biochemistry, molecular biology, genetics over the past century. It turns out that biology is a decipherable, programmable, in some ways, even digital system. Protein language models have demonstrated an astonishing ability to intuit the complex patterns and interrelationships between protein sequence, structure, and function. How changing certain amino acids in certain parts of a protein sequence will affect the shape that the protein folds into. And then he goes on to say that using AI platforms like ESM2, ESMFold, we can, quote, generate totally novel protein sequences that do not exist in nature. An opportunity exists for us to improve upon nature. After all, as powerful of a force as it is, evolution by natural selection is not all-seeing. It does not plan ahead. It does not reason or optimize in top-down fashion. It unfolds randomly and opportunistically, propagating combinations that happen to work. Using AI, he says, we can for the first time systematically and comprehensively explore the vast uncharted realms of protein space in order to design proteins unlike anything that has ever existed in nature. Unlike anything that has ever existed in nature, purpose-built for our medical and commercial needs. And you can see here that we run straight into two schools of thought, post-humanism and transhumanism. Post-humanism seeks to decentralize or displace anthropocentrism, the centrality of the human subject in the natural world. Transhumanism moves this several notches higher, where through genetic engineering, we are producing new kinds of proteins that never existed. And the implications are enormous in the sense that we are looking at creating a different kind of natural order, the natural world, and we're looking at a new kind of human subjectivity. So our notions of humanity, our notions of humanness are going to be up for grabs. So there is a lot at stake, and that's one of the key reasons why I titled the presentation, why bioengineering needs the humanities, excuse me. I'll pick up on 3 issues. 1, the philosophy of science. 2, CRISPR-Cas9 technology. And 3, the ethical questions raised by advances in bioengineering. So the philosophy of science, CRISPR-Cas9, and ethical questions. The first is the philosophy of science. What is the philosophy of science? Do humanists need to bother with it? Do scientists need to bother with the philosophy of science? Janet Stembedel addresses these questions in her article titled, "What is Philosophy of Science and Should Scientists Care?" It is possible for good science to happen and good scientists to conduct great science without bothering or worrying much about the philosophy of science. This is because the philosophy of science does not address specific activities, whether of a biologist, engineer, or entomologist. Rather, it concerns itself with science as a specialized endeavor, seeking, quote, to construct a picture of the world that is accountable to the world in a particular way. How that picture of the world develops and changes in response to further empirical information, and what kinds of explanations can be given for the success of scientific accounts, end quote. That means then the philosophy of science helps us to understand how knowledge is organized in a variety of research settings. The philosophy of science helps us to understand the protocols and expectations that shape the scientific endeavor. The philosophy of science sheds light on the nature of evidence. What kind of evidence do we need, and what kinds of scientific claims can be made based on what kind of evidence? And all said and done, the philosophy of science sheds light on science as a social endeavor, one that is supported by and oriented toward publics beyond the research center, beyond the university. So the philosophy of science, to quote Stembedel, is aimed at answering different questions that the working scientist is trying to answer. The goal of philosophy of science is not to answer specific questions, but to answer questions about science. This is an angle that Philip Kitcher takes up in his book, Science, Truth, and Democracy, where he talks about how we need to connect scientific value to political, cultural, and communal values. That's why he titles his text, Science, Truth, and Democracy. I can come back to Kitcher's proposal to develop a well-ordered science, probably in the Q&A. But let me move from Kitcher to Helen Longino, who, in her book, Science as Social Knowledge, Values and Objectivity in Scientific Inquiry, explores this issue further. She talks about the general tendencies, on the one hand, to view science as leading us to some kind of reliable objectivity and value neutrality. And then you have the social constructionists who sometimes take 2 positions. One is more moderate, in that they say that the scientific endeavor is constantly subject to political and social and personal agendas and a variety of interests. And therefore, there is going to be a lot of prejudicial or built-in agendas in any scientific endeavor, which need to be recognized and which need to be accepted and which needs to be addressed. Some social constructionists move to the extreme and say all that is true, which means that science is fundamentally contaminated, and we should eschew all notions of objectivity and neutrality. And Helen Longino raises these perspectives, but doesn't take that stance, and instead says we need to distinguish between constitutive values of science and the contextual values of science. And by constitutive, she means the goals of science, quote, to produce an explanation of the natural world using criteria as truth, accuracy, simplicity, predictability, and breadth. And by contextual values, she means, quote, the personal, social, and cultural values, those groups or individual preferences about what ought to be, those values that belong to the social and cultural environment in which science is done. So rather than looking at science and scientific discourse and the scientific endeavor as having the potential to be compromised, she reframes the debate to say, all knowledge, as a matter of fact, every kind of knowledge is subject to the same problems. So in that sense, science is not an exception to the rule, but it is part of the way things are done. And then she also says that by focusing on the constitutive values and the contextual values, we will see how we can, in many cases, contain or limit the problems of other interests seeping into science, because we can still go back to things like truth, accuracy, predictability, sharing of evidence, and transparency in the projects that we pursue. So we have Stemwedel's idea and Kitcher's text and Helen Longino, all three of whom are philosophizing about science. And which is an interesting thing, especially when Stemwedel says, this is not about individual scientists pursuing individual or particular scientific projects, but it is about the endeavor of science with a capital S. And that is where we can see a promising interface open up between bioengineering and the humanities, in this case, the field of philosophy. This takes us to the next topic, which is synthetic biology, which is about using engineering principles to develop microorganisms to modify the air, the water and soil and things like that, and picking up this language from the National Human Genome Research Institute. Other examples include genetically modifying rice to produce more vitamin A, to address the vitamin A deficiency. And there are other examples of making yeast to create rose oil that perfume makers can use rather than relying only on actual roses. And there are so many examples of synthetic biology. Let me pick up on two things that we talked about in our institute. One is the organoids, which are 3D cell cultures derived from pluripotent stem cells that mimic the structure and function and cellular complexity of human organisms. And then we looked at chimeras, which is about introducing human cells into other animal species. Host animals grafted with human brain organoids are a subcategory of brain chimeras. So we have organoids, the technology of making organoids, and the technology of introducing human genomic sequences in non-human tissue and in non-human organisms. So what happens when those kinds of research projects unfold, especially over time? Is there a point, for instance, in terms of values, is there a point where an organism should be treated as an entity with certain rights, just as we do non-human beings like animals? So what's the difference between animality and organism like lifeness, so to speak? Is that life? Or do we look at it in terms of its potential to simulate or become somehow human-like in some remote fashion? And does that potentiality itself give it a new status? So these are very perplexing questions that take us straight back to the underlying assumptions about science, and eventually they tip us definitely into the field of philosophy. So we looked at CRISPR-Cas9. We studied that fascinating technology, and we conducted a small experiment in the lab, a kind of a hands-on thing. So I will briefly describe what CRISPR-Cas9 is, and to folks who are new, hope this helps get a sense of the technology. So CRISPR refers to clustered, regularly interspaced palindromic repeats, which are genomic sequences that counter viruses. Cas9 is an endonuclease that cuts DNA at a specific site. So when you have CRISPR-Cas9, you have what they refer to as a molecular caesars, which enables a precise slicing of the DNA at a precise point in the sequence. There are other ways of gene editing, but what makes CRISPR-Cas9 dramatic and has galvanized all and led to all kinds of innovations in bioengineering is its simplicity and its precision. It combines simplicity and precision. So for our experiment, we did a blue-white screening of E. coli bacteria. So E. coli bacteria has the lac operon, which has the LAC-C gene, which codes for beta galactosidase. And this is where I'm probably messing up my pronunciation of these terms. So please bear with me. And I'm probably going to skip a couple of terms because I can't get the entire chemical order. So when it's referred to as BGAL, beta galactosidase, is active in bacteria, BGAL cleaves XGAL, which is a substrate, and which creates galactose and another substance that expresses a phenotype, and that phenotype is blue. So what we did is introduced plasmids into in a couple of bacteria cultures. And the plasmids had Cas9 technology, and they went and cut, or they interfered with that gene and knocked off its ability to express the blue phenotype, which in essence means BGAL is not cleaving XGAL, because when that happens, inevitably the phenotype is manifest. So we turned that off, and in another bacteria culture, we didn't turn that off. And then we put that into, we did the shock treatments and the ice treatments and incubated them for about 24 hours. And we went back with bated breath after doing all of these experiments, and to see how one set of bacteria culture manifests the phenotype and the other doesn't. It's a relatively simple experiment, and probably our colleagues in microbiology would say it is. But to us, it was an intriguing way to do a hands-on experiment to see how we can go into the details of molecules and enzymes and start turning on and off certain things. And the implications, for instance, of doing this kinds of genetic editing are enormous. They raise so many ethical, political, and other questions. So this is about CRISPR-Cas9. A kind of an FYI, Jennifer Doudna, a scientist in the US, and Emmanuel Marie Charpentier from France, they had began partnering in 2011, and they eventually ended up perfecting this gene editing technology. And they were awarded the Nobel Prize in Chemistry, I think in 2020. The first time a Nobel Prize was shared by two women. There is a book by Walter Isaacson called The Codebreaker, Jennifer Doudna, Gene Editing and the Future of the Human Race. I have heard Isaacson talk about this book and some videos, and I'm looking forward to reading it. Isaacson is a good biographer. He has written on Steve Jobs, more recently last year, I think, this year, on Elon Musk and Leonardo da Vinci. it's a probably a good book to get into. So let me end with one example of a question that all of us discussed in the institute, and how it can shed light on the interfacing of bioengineering and the humanities. So we have CRISPR-Cas9's broad applicability, and then we have two possibilities. One is gene extinction or species extinction. So if we look at the Anopheles mosquito or the marsh mosquito that causes malaria, using a deliberate extinction as a model, we can target this mosquito for eradication, just as we are looking at targeting the screwworm and other species of rats and mice. But this raises ethical questions. Should we completely eradicate the Anopheles mosquito because it is prone to carrying the vector, prone to becoming a vector of Plasmodium, the parasite that actually causes malaria? So the other, not a solution, but another approach is the gene drive, which is to modify the gene so that the mosquito we knock off the ability of the mosquito to become a vector of Plasmodium. In other words, the mosquito continues to exist. We're not driving it into extinction, but we're trying to limit its ability to carry or become a vector for malaria. So when we're looking at, when we frame these kinds of questions in this way, we're looking at the instrumental value of science in that context, in the sense that, is this going to benefit certain people living in a certain subtropical or tropical area? Are hundreds and thousands of people going to be prevented from being exposed to malaria, and many of them even getting it? Another way of framing this is to look at what role the Anopheles mosquito plays in the ecosystems of particular regions. And when we look at when we use an ecosystem as a frame of reference, then inevitably we have to engage with the kinds of interrelationships among so many living things and organisms. So that reframing of the question then led us to ecosystem examinations and environmental issues in terms of how our engagement with particular gene editing technologies led us to continually, in a way, broaden the scope of philosophizing about science, particularly in terms of the ethical dimensions of bioengineering. So to sum up, we looked at the AI and protein language models, the philosophy of science, and CRISPR-Cas9, and the ethical challenges of genetic engineering. And I offer these comments as a way to hopefully initiate conversations and listen and learn. And given the fact that we are now with unified accreditation as a model for the humane system, where hopefully we can engage in collaborations of all kinds. So thank you for the opportunity, and thank you for listening. I'll open this up. Yes, Danielle.
That was fascinating. My brain's clicking away, thinking about all these really interesting collaborations, and I'm already thinking of how I want you to potentially do a training for our researchers around these topics, because I think it'd be really powerful. But anyways, please, if anybody has a question, you can unmute yourself and ask, or if you want to engage in with a comment and start a conversation amongst the group, or again, you could throw it in chat. And while we wait to see if anybody besides me wants to ask a question, I will go ahead and get started, because I've got some written down here. First of all, how did you hear, and I'm going to bring us back away from the deep dive into the content itself and the actual process, because a part of having you come to speak, besides in initiating these conversations and these thought-provoking pieces, is to try to encourage other folks to consider this program as a potential. And my question to you is how did you hear about it? And was this a topic that you defined for your experience, or was it an experience that you chose to participate in?
I have been involved with at the system level. I had the wonderful opportunity of working with a host of people across the system last year on drafting the strategic plan, and ended up getting in touch with folks, and I made a few visits to the Orono campus, and am the affiliate graduate faculty. I think that's the designation I have with the program in intermediate studies, which I think is now housed in the Department of Art. So I've met with a couple of folks and students in that program. And of course, Jason Charland has been, he leads the research endeavor there. So on and off, we were talking about these things, and we were looking at how, for instance, in the strategic plan, we have certain areas that have been highlighted for potential investment. And those areas include artificial intelligence, bioengineering, and the digital humanities, and some other fields that can hopefully enable faculty across the system and researchers across the system to pursue collaborative ventures. So that was my exposure to a lot of the work that is going on at UMaine Orono, and I got the invitation to speak. I was on a sabbatical last year, and I've been doing a lot of work in the digital humanities. And one of the things that always struck me is the liberal arts education model. Our presidents, provosts, national academies of science, of the humanities, we all regularly and necessarily and understandably engage in generating the rhetoric about the liberal arts. And what does it mean? It means essentially drawing from multiple streams of knowledge. That is the raison d'etre of the core curriculum, as a matter of fact, the general education program. And so when we ask our students to use your critical reasoning skills, to pursue your curiosity, to see where it leads, and don't worry about failing, understand how different branches of the knowledge are branches of the same tree. I was thinking to myself, and I had enough time during the sabbatical, and when I came across the NEH announcement, that sounded intriguing. And so I reached out to Doctors Robert and O'Neill and asked them, this is what I'm doing at USM, and I have no idea about bioengineering, but this is what I can bring to the table as a humanities scholar, now also addressing the intersection of digital technologies and the humanities. Would you give me shed some light on what you think I could do, and if someone like me is even a good candidate? And they responded and said, we're looking for the kind of folks who are doing what you were doing in terms of how humanists can integrate science into humanities courses or humanities-oriented research projects. So that is how we ended up at ASU, and we had a wonderful session at Arizona State University. It's an amazing institute, and we had a great session for 4 weeks. I hope that explains how I happened to come about it.
Yeah, that's great. What would you say, and you've alluded to this already, at least from the standpoint of how it fits into the strategic plan for research and development across the system, but outside of that, what would you say was the ultimate goal for you to participate in this? And I don't want to lead you down one path or another. So I'll leave it at that.
My goal to put many in terms of why I'm doing this. Jason Robert, one of the directors, has an interesting essay. He talks about his essay is titled "Toward a Better Bioethics: Commentary on Forbidden Science, Some Beginning Reflections." He talks about how these perceptions about the humanists and bioethicists who are trained in humanities fields, sometimes they can be viewed as the morality police, sometimes as people who are waving the cudgel of moral responsibility and trying to rein the disorganized or excessively ambitious scientists. He says those are very problematic and narrowly conceived ideas. And he says we can think of our roles as architects of a moral space. So we are developing a moral architecture in which many of these issues and questions can be raised. It's not about playing the role of the arbiter or judging things. And he does say that for us to engage in creating this kind of space, anyone who wants to do this and be part of it, you have to learn the language of science. So to me, one challenge was, the biggest challenge was learning about biology. All of us at the Institute had not entered a wet lab for 35, 40 years. And so we went through an entire weeks of orientation. They gave us lab coats. Oh, it's hanging there proudly in the closet, and I hope I can wear it. But it gave us that experience of what scientists do, which is so different from what I do regularly, teaching and writing about texts and literature and things like that. That was an eye-opening and a wonderful experience offered by the National Endowment for the Humanities and, of course, Arizona State University. So I would like to move in that direction, Danielle, to enact what we tell our students often, because as scholars, our responsibility is not only to provide pathways for other researchers and students, but to actually trudge those pathways too. And unless we do that, unless we can demonstrate that we have a stake in it, and it won't help. It's just going to be rhetoric in terms of just the nice flourish of a garnishing of ideas, things like that.
Was there reciprocity there in terms of the scientists learning about how you all think, and you were talking about how you all had to learn the biology language. Was there learning on the other side of that as well?
Yes. We met with some great folks. I think I can mention this to you. The former president of the American Association of Biological Sciences, Jim Collins, he came and spoke to us. Both Jason Robert and Erica O'Neill organized a great slate of speakers. So we're getting a director of this program, a director of sustainability, the vice provost for innovation. So we had a great lineup of speakers. And what stood out to us was that almost every person who spoke to us were not speaking to us as people coming from the sciences, but were already familiar in a way. They had a humanistic sensibility, let's put it that way. And what stood out, all of us were amazed by this. And it turns out that ASU, over the last 20 or so years, under the leadership of its president, Michael Crow, who is still the president now, embarked on a series of institutional initiatives. And the key focus there was in bringing faculty and researchers together, getting the administrators out, putting them together, incentivizing them, and asking them. And if a group of faculty would go to a dean and the provost and say, this is what we think we need to do for the next 10 years. Instead of having, let's say, a department of engineering with a couple of bioengineers in that, why not form a school of bioengineering? We'd have a person from philosophy, from literature, and 2 or 3 people do from molecular biology and microbiology. And we could come up with a school and move on from there. And that has been, or is, probably a signature strategy of Arizona State University. So that stood out to us in terms of institutional cultural transformation and its wide effect across the board, and in terms of the interactions we had with particular individuals, where we could see that they already had a considerable exposure to people from other disciplines. And it was manifest in the way they spoke and the way in which they would raise cross-referential terms and examples.
Very neat. All right, so there's a question in the chat. Was this NEH conversation informed by adaptation and natural selection, concept authored by George C. Williams in 1966, or the selfish gene, authored in 1976 by Richard Dawkins? Well, we have somebody who knows their stuff here. Or the revolutionary phenotype, authored in 2018 by J.F. Gariepy. These concepts build on each other precisely in the direction on this topic. Wow.
Yes, we mentioned the selfish gene, and thank you to the person who is mentioning these texts, because those are the texts that would be useful to read up on. And if you can post it in chat, or if you can email some of this information to Danielle, and if Danielle can send that over to me, that'll be great. I would love to connect and talk more about this. As a matter of fact, I should mention this to you. I did suggest to Jason Sharland the possibility of having or convening a group of faculty and researchers from UMaine and from different universities in the UMaine system, so that we could hear what other folks are doing, and more to the point, see if there are overlaps and opportunities to push a couple of questions further as research agendas and see what may ensue. So thanks for the references.
Jonathan, would you mind unmuting yourself? I'm just curious of what your background is.
Mostly structural engineering and construction, but I've had a fair amount of experience working with biotech, and that has allowed for a lot of interesting cross relationships and discussions.
Sure.
With those former colleagues.
And are you part of the University of Maine system?
Yes, ma'am. I work with the physical plant and the facilities and general services departments.
Wow, very cool. What a great question. Great resources.
Yes, thank you.
Maybe I'll jump in with a question. There was a phrase you used when you were talking about the philosophy of science that I was wondering if you could elaborate a bit more on, particularly in these very or interdisciplinary discussions, such as at the Institute, about the nature of evidence, just because it takes a very different approach to change your mind based upon evidence, when we do, coming from very different disciplines. So to have a sort of a collective understanding of what does constitute evidence, was that part of the discussion? Was that part of the orientation for your institute? And how did that go?
Yes, and all of us were humanists, and we were trained in reading between the lines, so to speak, a phrase that is common for humanists and generally in higher education, to read between the lines, because it is text oriented and you're picking apart the implications of a particular syntactical structure or linguistic arrangement, as it were. But in terms of the kinds of claims that could be made, what is the nature of evidence? One thing that often came up, of course, was the COVID pandemic, how this disrupted the entire world, and even to date, how there are so many intense debates about the causes of the pandemic, the solutions of lockdowns, the way in which the economic sectors were impacted and the educational sectors, and what kind ultimately of evidence that policymakers had to exercise that authority to move the entire country and the entire world in so many ways along particular directions. And we're still battling those out. Those things are not settled. And so we ended up looking at how evidence by itself is not constitutive of legitimacy. So evidence, textual evidence or data or anthropological evidence or scientific evidence, they are woven into certain protocols and organizational procedures that give them the social and political and economic legitimacy. When we remove those strands that bind evidence, let us say, to the organizational protocols, to the disciplinary protocols, when we remove all of those strands, it amounts to just evidence, which is just a data set. So that was a reframing that we had to do, and Kitcher says that how a scientific value gets linked to economic value and political value, not because of something inherent in science or inherent in evidence, but because of the discursive structures in which evidence gets played out and connected to a range of other discourses that have a legitimizing function in society, a legitimizing function. So that's one of the discussions we had, Ed. I hope that sheds light on at least one aspect of your query.
No, that's excellent. Really very helpful, because it leads to the question again, coming back to policy and law, because we may not be, as a bioethicist, one may not be the morality police, but we are police. When we set policies based upon the principles of the president at the time, say if you're looking at stem cell policy, and how that can whipsaw, then there is this the connection becomes from the legitimacy comes not from scientific evidence, but from some other basis, and so from somebody placing themselves as the moral police. So it's kind of your explanation of these discursive structures makes it clear where what strands connect to legitimacy can be very different than where somebody from the science field might think it would have started from scientific evidence, but rather all these other strands can be a lot stronger. So that's I think that really helps me understand a bit better those connections. But maybe more about policy and law and how all this connects. Maybe that also came up in the workshop or in your work.
Yes. And if we also look at globalization and the unevenness that globalization has generated over decades, and we're looking at different kinds of scientific cultures in different nation states, and the culture of free expression and the scientific enterprise that we have in developed societies like the United States are quite unlike other cultures in other countries. The other thing that also came up with, and I think I could quickly comment on this, which was an intriguing thing that Jim Collins shared with us. In 1990, I think the National Academy of Science published a report called the Liberal Art of Science in 1990. It talked about the importance of scientific literacy, particularly at the undergraduate level. So Jim Collins shared with us a proposal that he and his colleagues wrote and submitted for funding. He shared with us, but I can't share that with you because it's still under review. But we talked about how from so many things have happened since 1990, and one of the biggest disruptive technologies has been the digital technology. And we've had the pandemic, and we have greater public questions about the value of higher education. So the three issues that he shared that we discussed that I could probably share with you, because they were thinking of convening a group of scientists and educators from across the country to issue another report called probably The Liberating Art of Science. So in 1990, it was the Liberal Art of Science, and now it is going to be the Liberating Art of Science that would focus on critical reasoning and evidence analysis, and interestingly enough, creative intelligence and problem solving, and addressing issues of inclusivity and public relevance. Just as an FYI, in terms of the broader organizational and institutional reforms that might be initiated by a range of scientists across the country that speak to many of the issues that we just pointed out.
We have Dave Gustin on that as well.
Yes, we actually had Dave Gustin talk to us. We had 2 sessions with Dave.
Okay, very good. Yeah.
Do you know him?
Yes. I used to interact with these guys a number of years ago. Yeah.
Okay. Were you at ASU too?
No, I was at AAAS. And so Dave Gustin was on an advisory board there, the Committee on Science, Engineering, and Public Policy.
I see.
And Jim Collins served on a panel that I managed once upon a time as well. So
terrific.
Yeah.
Oh, great. Small world indeed.
Well, my question, kind of bring everything sort of to a conclusion here, is best practices, strategies to start or continue these authentic conversations across disciplines, where you do speak different languages and have different approaches. What do you think is sort of, how can we keep this going forward? And not just on this topic in general, but, the interweaving of arts and humanities and sciences, and actually all, really. I think everybody, having those voices around the table is important.
Mm-hmm. You mean how we can extend the work that we're doing, Danielle?
Or just to start these conversations with, bringing new people into the conversation, and or working towards a common goal. I mean, how do you start those conversations with people? Do you have, what is, a strategy or strategies that you would recommend?
Can we turn the recording off?
Well, that's interesting.
I don't want all of us to get into a lot of trouble or something. I'm just kidding. But in a way, I'm not too, because these are difficult conversations we all need to have.
Yep.
And maybe we can edit this later if necessary, but we need to have administrators, provosts, deans, members of the board of trustees, presidents, rethink leadership models. Rethink leadership models. So we have probably a lot of people can relate to this, where rather than convening the researchers and faculty members and the ones who are engaging in these kinds of discussions, we can have leadership structures in which deans or presidents or provosts or directors over here, they get to decide what matters, and then investments follow. But rarely do we have the administrators explaining to us the intellectual relevance of what they want to do. And that is, it's a leadership issue too. I'm just sharing some things, reflecting also. And what struck us about what happened at ASU over 20 years, and ASU now is competing with the big entities like Stanford and MIT and Harvard. And the little, of course, ASU also has a lot of problems, no doubt, and it's not the only one that is doing some wonderful work. But what struck me about the leadership strategies that President Michael Crow was advocating for and practiced was how the administrators can conceive of their roles as facilitating spaces for intellectual fermentation to take place, rather than weighing in and limiting the conversations and the work that is being done, and then orienting the entire university toward a corporate kind of mentality, which is driven primarily on efficiency and market value. And as a humanist, I am not saying, and I will not say, that we don't need to discuss those issues at all. We have to. And God knows we are seeing precipitous drops in English majors across the board in many universities in the country. So we have to pay attention to market relevance. We have to pay attention to finances. But within the larger context of driving the humanistic enterprise of knowledge generation and learning. And it'll be great to have spaces like this where we can convene and meet and not be, sometimes conversations like this may not go somewhere, and it is okay, because the efficiency model says it was a waste of time. Why did we have so many people come? Why did we do this? But if we structure it in a way where we understand that one of the limitations of this kind of experimental work is that you realize that maybe that is not the position or the trajectory you want to take, that is a good learning outcome. That's the kind of rethinking of leadership strategies and models that probably we can consider.
Well, that's great. I mean, it's like you mentioned way at the beginning of your talk, we're sort of in a prime position to start thinking about this sort of holistic or system culture about these things, with that unified accreditation and, continuing these conversations on interweaving, and these transdisciplinary approaches. Wow. Well, my brain is going, more than anything else, even if it's just a raising of awareness and starting conversations with the people that were able to be here today, and hopefully the people that will take the time to watch the recording of this, because we will post it. I mean, wow. Thank you so much, John, for sharing your experiences with us and your passion. I mean, it's palpable. I really, really appreciate it. Everybody take a look at the ORD events page. Our next month's presenter will be a week off because the 1st of January is obviously the first Monday in January is the holiday. So it will be on January the 8th. And Babak Kajradi from Maine will be talking about his experiences with NSF career, as an NSF career awardee and an NIH awardee on topics of wearable robotics for gait training. So please check that out. Register if you'd like to attend. And John, thank you again. I appreciate it. And everybody who joined us today, have a wonderful day.
Thank you very much, Danielle and Tammy. And thank you to everyone who took the time to attend the webinar. Thanks a bunch.